All right, bubble up, everybody, because today we're diving into a world that's usually completely invisible to us.
We are.
Yeah, we're going to explore the world of single molecules.
Oh, wow.
You know, you sent me some of the research on single molecule tracking,
Right.
SMT,
SNT, yeah.
and honestly, I'm a little obsessed with the idea that we can actually see these molecules in action.
Yeah.
We're talking about seeing how viruses, try to take over our cells and how our brains work at like the most fundamental level.
Right.
Even how all of this is changing the way new drugs are being discovered.
Yeah.
It's like we suddenly have, microscopic superpowers.
It really is kind of revolutionary because for so long we were only able to look at molecules in, huge groups.
Mhm.
And we got the sort of average picture of how they behave.
Right.
But with SMT, we can actually zero in on individual molecules.
Wow.
And that's where things get really interesting.
Okay, so we're not looking at the whole crowd anymore. We're
No.
focusing on like the solo artist.
Yeah,
But
exactly.
why is that so important?
Well, think about it this way. Imagine you want to figure out how a city works,
Okay.
but you can only see it from a helicopter, like the traffic flow.
Okay.
You miss all the individual interactions, all the deliveries,
Right, yeah.
people going about their lives.
Right.
S&T is like, zooming right into street level and seeing how each molecule interacts with everything around it.
So we're finally getting like the personality of the city,
Yeah.
not just like the overall, vibe from a distance.
Exactly.
And you're talking about these individual interactions, and it reminded me of something that was in the research that you sent over.
Okay.
Like it turns out that all molecules, even if they're the same type, they're not all created equal.
No, not at all.
Like they have their own little quirks, just like people.
They really do.
Okay.
And that's one of the big things that's come out of SMT, this idea of heterogeneity.
Yeah,
Right?
I remember that word.
For example, in that research on Enzyme X, SMT showed that only a small percentage of those enzymes, they actually adopt the right shape
Oh, wow.
for catalysis, for their actual job.
Huh.
And that's something you would totally miss if you were just looking at the average behavior of a big pool of them.
So it's like, okay, you have this whole keychain full of teams,
Right.
but only a couple of them actually work.
Exactly.
Even though they all look identical.
Right.
This is really amazing.
It gets even better because imagine like you're trying to study a dance,
Okay.
but you can only see this blurred like movement of the dancers.
Uh-huh.
With S&T, it's like we can slow down the music and we
Right.
can zoom in and see every single step, every interaction.
Wow.
And that's the power of this thing called temporal resolution. It lets us catch those really, really fast, fleeting moments like one molecule binding to another one.
Okay.
Things that you would completely miss with traditional methods.
It's like having a high-speed camera, but for the molecular world.
Exactly.
And, that's just the beginning, right?
Right.
Because then there's also this thing called spatial resolution.
Exactly.
And it's like being able to zoom in to the nanoscale,
That's amazing.
which is just wild to me that we can actually pinpoint the location of individual molecules with such incredible precision.
Right.
But how does all of this actually translate into, real knowledge? Like, what are the secrets that this molecular close-up is actually revealing to us?
Well, think of it this way. Like, imagine that you want to see those individual proteins that make up the shell of a virus. It's called the capsid.
Oh, okay.
With these techniques like this thing called STORM, which is a type of super resolution microscopy.
Okay.
You can map out where every single protein is, and you can create this 3D model of this incredibly tiny, tiny structure.
Oh, wow.
It's like going from a blurry photo to this, high definition blueprint.
Wow.
Yeah.
Okay, that's a game changer.
Yeah.
It's not just seeing where things are, but like how they fit together.
Exactly.
How they work is this whole system.
Right.
But before we get totally lost in all the amazing things that we can do with SMT, can we take a step back for a second
Sure.
and kind of walk through how it all works?
Yeah.
Like, I'm picturing it as, kind of a journey. We start by tagging a molecule, and then we end up with some kind of, mind-blowing analysis.
That's a great way to think about it.
Yeah.
Yeah. So first things first, we need a way to actually see the molecule.
Right.
Right. It's like you're trying to follow one specific ant in a giant colony.
Oh, yeah.
You've got to mark it somehow so you can see where it goes. And that's where this thing called labeling comes in.
So we're giving our molecule like a little GPS tracker.
Exactly.
I love that.
Yeah.
So tell me more about these labels.
Well, there are a bunch of options, and each of them has, its pluses and minuses.
Okay.
One of the most common ones are these things called fluorescent dyes.
Okay.
They're like these little tiny light bulbs, and they give off light when you shine a laser on them,
Okay.
and they come in all kinds of colors, so you can even track multiple molecules at the same time.
Wow, so we've got our molecule all lit up.
Yeah,
It's ready for its close-up, but what kind of microscope do we need to actually see this thing?
that's a good question.
Because we're talking about, like, the molecular level here. We need some serious magnification power.
We need a lot of magnification. So the type of microscope, you know, really depends on what you're trying to see.
Okay.
So if we want to see things that are happening really, really fast, like those molecules binding,
Right.
we might use something like turf microscopy, which illuminates just a really thin slice of the sample,
Okay.
and that cuts down on the background noise, and it's really good for studying things like cell membranes.
So turf is like shining a spotlight on the state.
Exactly. Yeah.
It's focusing our attention where it needs to be.
Right.
I remember earlier you mentioned this thing called super-resolution microscopy.
Yes.
Now that's where things get really wild.
That's where it gets really exciting.
Right.
Yeah, techniques like STORM and PALM, these techniques let us break the limits of normal traditional light microscopes.
I see.
So to get a sense of how powerful this is, imagine you're trying to read a book,
Okay.
but all the letters are just blurred together.
Right.
Super resolution microscopy is like putting on these magic glasses that
Right.
suddenly make all those letters come into focus.
Like going from standard definition to 4K.
Exactly. Yeah.
And I'm guessing to capture these incredible images, we need some pretty special cameras as well?
We do. We need some really, really fast cameras. High-speed cameras are super important for SMT
Okay.
because they can record, thousands of frames a second.
Wow.
And that allows us to see those super, super fast molecular motions that would otherwise be invisible.
Okay.
It's like watching a hummingbird's wings in slow motion.
Okay. So we've labeled our molecule. We've chosen our fancy microscope. We've
Yeah.
captured its movements on camera. Now we have all these images, all this data.
Yeah.
What happens next?
Well, that's where the real detective work comes in, right?
Right.
We've got to take all those microscope pictures and we've got to translate them into like a molecular roadmap
I like that.
that shows us, where our molecule went and what it did over time.
Okay.
This means we have to pinpoint where the molecule is in each frame,
Which is-
called localization.
Got it.
And then we connect all those different positions across all the different frames
Okay.
to create what's called a trajectory.
I see.
Its path.
So it's like we're connecting the dots to reveal the secret journey of this molecule.
Exactly.
I love that.
Yeah.
So what can we actually learn from this molecular roadmap?
Well, that's when the analysis gets really interesting, right?
Okay.
So we can calculate things like, diffusion coefficients,
Okay.
which tell us how fast the molecule's moving.
Right.
Or we can look at this thing called mean square displacement, which measures how much it's exploring the space around it.
I see.
And if we see sudden changes in these values, it might mean that our molecule is, bumping into something else, like
Wow.
another
Wow.
molecule
Oh, wow.
or a cellular structure.
So we're actually seeing these interactions happening in real time.
We are.
That's amazing.
It is. And we can actually go a step further and we can analyze conformational changes.
Okay.
And that means that we can see if the molecule changes its shape over time.
Okay.
And this can tell us a lot about how it works,
Right.
especially for proteins, which often need to change shape to do their job.
It's like watching a tiny origami creation folding and unfolding.
Exactly.
I love it. But I know this is just the tip of the iceberg.
Oh, yeah.
SMT isn't just about watching molecules move around. It's about using all of this information to understand how these real-world biological processes are actually working.
That's what it's all about.
Right.
We can use SMT to study everything from how proteins are interacting with each other inside our cells
Right.
to how viruses try to hijack our cellular machinery.
Okay, I'm officially hooked. Let's dive into some of these specific applications.
All right, let's do it.
Awesome. Okay, so before we got sidetracked, we were about to explore some of the amazing real-world applications of SMT.
Right.
Where do you even begin with something this powerful?
Well, a lot of the research you sent in focused on proteins.
Right.
And that's a great place to start.
Okay.
Proteins are like these tiny little machines that keep our cells running.
Okay.
And with SMT, we can actually watch them in action.
It's like having a backstage pass to the most intricate show on Earth.
Exactly.
So give me an example.
Okay, well, let's take receptors on cell membranes.
Okay.
These proteins act like gatekeepers.
Right.
They control what can go in and out of a cell.
Right.
And for a long time, we thought that they were just kind of stationary, but
Oh, okay.
SMT has shown us that they're actually constantly moving around.
Don't they?
They interact with other molecules. They even, cluster together in groups.
So it's not just a gate.
No.
It's like a whole security checkpoint.
Right.
You've got guards moving around, checking IDs, directing traffic.
That's a great analogy.
I like that.
And this dynamic behavior, it's so important for how cells communicate with each other
Okay.
and how they respond to what's going on around them.
Right, right.
And it's also really crucial for understanding how drugs work, because a lot of drugs actually target these receptors.
Right. So instead of just assuming that a drug fits into a receptor like a key in a lock,
Right.
we can actually see this whole interaction play out in real time.
Yeah.
See how the receptor moves and how the drug is influencing that movement.
Exactly.
Okay.
And that level of detail, it's really valuable for drug discovery.
I bet.
But, receptors are just one example.
Right.
We can also use SMT to study how motor proteins walk
I love that.
along microtubules.
Oh, yeah, the cellular highway system.
Exactly.
I remember reading about that in the research you sent.
Yeah.
It's just fascinating how these tiny proteins can carry cargo that's many times their own size.
It really is. And with SMT, we can actually track these motor proteins step by step. We can see how they bind to the microtubule, how they use energy to move, and how they release their cargo when they reach their destination.
Okay.
It's like watching a microscopic delivery truck navigating through a busy city.
That's a great analogy. I love it. Okay, I'm really starting to see how SMT is changing, the whole game in biology.
Yeah.
But what about DNA and RNA?
Oh, yeah.
Can we use SMT to study these molecules that actually carry our genetic code?
Absolutely. SMT has been a real game changer for understanding how DNA is replicated, how it's repaired,
Right.
and how it's transcribed into RNA, which is the molecule that carries the instructions to make proteins.
So we can actually watch this flow of genetic information
Yes.
from DNA to RNA to protein.
You got it.
It's like seeing the blueprint of life come to life.
That's the beauty of SMT. For example, we can track this enzyme called RNA polymerase as it moves along a strand of DNA
Okay.
and transcribes the genetic code into RNA.
Okay.
We can see where it pauses, if it backtracks,
Wow.
and how it interacts with other proteins that are regulating the process.
So we're like listening in on the conversation between DNA and RNA.
Yes.
Wow.
Understanding how genes are turned on and off.
Okay. I have a feeling, though, that you've saved the most dramatic application for last.
I might have.
Tell me about how SMT is being used to study viruses.
Okay, well, viruses are fascinating.
Uh-huh.
They're also a little bit scary. They're basically these tiny little packages of genetic material
Right.
that hijack our cells in order to replicate.
Wow.
And SMT has given us this incredible, unprecedented view of this whole process.
It's like watching a microscopic heist unfold.
That's a great analogy, yeah. So, for instance, we can track individual virus particles as they enter a cell.
Okay.
We can see how they bind to receptors on the surface of the cell.
Right.
And we can even see how they release their genetic material inside the cell.
Wow.
We can even track those newly made viral proteins as they assemble into new virus particles.
So we can see the whole life cycle of the virus from infection to replication.
Exactly.
That's amazing.
And incredibly important for developing, antiviral therapies.
Right.
So, for example, in that research that you sent over, they talk about using SMT to study how certain antiviral drugs block the entry of viruses into cells.
Oh, right. Yeah.
And by tracking the individual virus particles,
Yeah.
researchers could actually see how these drugs interfered with that binding process.
I see.
Which gave them some really valuable clues for developing new drugs.
It's like we're watching a security system in action,
Right.
seeing how the virus tries to break in
Exactly.
and how this antiviral drug is acting like this, super strong lock to prevent the entry.
Yeah, that's a great way to think about it.
But I can imagine that studying viruses, the SMT, presents some pretty unique challenges as well.
It does. Viruses are incredibly small,
Yeah.
and they move really, really fast,
Okay.
which makes them really tough to track.
Right.
And because they replicate inside cells, we need ways to label them and image them in this
Right.
really complex environment.
So it's like we're trying to track this tiny, speedy thief in a crowded marketplace.
Exactly.
But I'm guessing scientists are coming up with some pretty clever solutions.
They are. They're very clever people.
Right.
So one approach is to use these fluorescently labeled antibodies that bind specifically to the virus proteins,
Okay.
and that makes them visible under the microscope.
Got it.
And another strategy is to actually label the virus itself with a fluorescent dye.
It's like we're putting this tiny little beacon on the virus so you can
Exactly.
follow its every move.
Yeah.
But, I remember from the research that one of the other challenges with SMT
Yeah.
is just dealing with this massive amount of data that these experiments generate.
It's true. SMT experiments generate a huge amount of data.
Right.
And analyzing it can be, pretty daunting.
It's like trying to make sense of a library full of books that are written in a language you don't even understand.
That's a great analogy. But luckily, we have all these powerful computational tools now that can help us
Okay.
sift through all that data and extract, meaningful information.
Okay.
So, for example, there are algorithms that can automatically track the movements of hundreds or even thousands of molecules at the same time,
Wow.
identify patterns in their behavior,
I see.
and even create 3D models of their trajectories.
So it's like having a whole team of superpowered research assistants who
Exactly.
are working 20/47
Yeah.
to analyze this data and kind of point out like the most important findings to us.
That's a great way to think about it.
Okay, so it's not just about the microscope. It's about this whole ecosystem of tools and techniques
Right.
that are pushing SMT forward.
Yes,
And this has been an incredible journey so far.
it has.
And I'm eager to see what the future holds for this revolutionary technology.
Me too. But, before we get to the future, I think it's worth
Yeah.
kind of taking a moment to acknowledge some of the challenges that are still out there.
Okay, let's get real for a second. What are some of the hurdles that scientists are still facing with SMT?
Well, one of the biggest ones is this thing called photobleaching.
Okay.
And that's the tendency for those fluorescent labels to fade over time when they're exposed to light.
Okay.
And this can really limit how long we can track a molecule. And it makes it harder to study those slow processes or the events that don't happen very often.
It's like you're trying to follow a trail of breadcrumbs that just keeps disappearing.
That's a good analogy.
Right.
Another challenge is this thing called photoblinking.
Okay.
That's where the fluorescent signal kind of flickers on and off, and that
Yeah.
makes it really hard to track the molecule's path accurately.
Oh, okay. So it's like trying to read a book with a flickering light.
Exactly. But don't worry, scientists are clever people.
I'm sure they are.
They're always developing new strategies to overcome these challenges.
Okay, I'm all ears. Tell me more about how researchers are tackling these issues head on.
All right, let's do it.
Okay, so we were talking about some of the challenges of, working at this molecular level.
Right.
Like photobleaching and photoblinking.
Right.
I'm ready for some solutions. What are scientists doing to kind of tackle these issues?
They're coming up with all sorts of clever approaches. So for photobleaching, imagine you're trying to read a book, but the light's fading.
Right.
You want a brighter light source.
Yeah.
Well, researchers are developing these new fluorescent labels that are brighter and more resistant to fading.
Okay.
It's like, going from a flickering candle to a powerful LED.
Oh, I like that.
Yeah.
So stronger, longer lasting labels.
Exactly.
That makes sense.
Yeah.
What about the flickering issue, the photoblinking?
Yeah.
I remember reading about quantum dots in the research that you sent.
Right.
Are those helpful here?
They are.
Okay.
Quantum dots are really great because they're super bright and they're very stable,
Okay.
which makes them perfect for long-term tracking.
Okay.
And in fact, in that research you sent, they used quantum dots to actually track the movement of receptors on immune cells,
Wow.
which gave them some crucial insights into how those cells communicate.
Okay.
But like any tool, they have their limitations.
Right.
Quantum dyes are a little bit bigger than traditional dyes.
Okay.
And sometimes that can affect the behavior of the molecule you're attaching it to.
Right. So it's a trade-off. Like choosing between, a nimble scooter
Yeah.
or a sturdy truck, depending on what you're trying to transport.
Exactly.
But it sounds like, the toolbox is constantly expanding.
It is.
So what other tricks do scientists have up their sleeves?
Well, on the microscopy front, techniques like turf microscopy are really helpful.
Yeah.
Remember how we talked about that spotlight?
Yeah.
So by illuminating just a thin section of the sample,
Right.
it reduces how much light the molecules are exposed to,
Okay.
which, in turn reduces photobleaching.
Okay, clever. It's like we're protecting those delicate molecules from the glare of the spotlight.
Yeah.
But even with all these advancements, we're still dealing with these mountains of data.
We are,
Right.
yes.
It's like trying to find a needle in a haystack of molecular movements.
That's where the power of computers really comes in.
Uh-huh.
Remember those sophisticated algorithms we were talking about?
Yes.
They're becoming more and more important for analyzing this SMT data.
Okay.
We're talking about algorithms that can automatically track the movements of thousands of molecules at the same time,
Wow.
identify these really subtle patterns in their behavior,
Okay.
and even create these 3D models of their trajectories.
So it's like we have a whole team of superpowered detectives
Right.
who are sifting through all the clues and piecing together the molecular story.
Yeah.
And I imagine artificial intelligence is playing a pretty big role in this as well.
Oh, absolutely. Machine learning is totally transforming the way that we analyze this SMT data.
Okay.
So imagine you're trying to identify a specific type of bird in a flock.
Okay.
A machine learning algorithm can analyze pictures of all the birds,
Right.
learn those tiny little differences in their appearance,
Yeah.
and then automatically pick out the bird that you're looking for.
So it's like training a computer to see the world through the eyes of an expert bird watcher.
That's a great way to think about it.
That's amazing. But even with all these incredible advancements, I'm curious, what are the big unanswered questions in SMT? What are the frontiers that scientists are still kind of trying to push?
Well, one of the most exciting ones is this thing called in vivo tracking,
Okay.
which means observing molecules inside living organisms.
Wow.
So this would take SMT to a whole new level because we could actually study these biological processes in their natural context.
It's like going from studying animals in a zoo
Yeah,
to actually observing them in the wild.
exactly.
Wow. So what are some of the challenges of in vivo tracking?
Well, living organisms are really complex environments, right?
Right.
They're full of moving parts and all sorts of background noise.
Right.
It's like trying to track a firefly in a thunderstorm. So we need ways to label and image molecules with really high specificity and sensitivity.
Okay.
And we need to come up with new computational tools
Right.
to make sense of all that data.
So it's this combination of pushing the limits of the technology and developing smarter ways to actually understand the information that we're getting.
Exactly.
It sounds like an incredible challenge.
It is.
But I'm sensing there's another frontier that's maybe even more mind-blowing. Multicolor imaging.
Ah, you read my mind.
Right.
Multicolor imaging is really exciting because it lets us track multiple molecules at the same time,
Wow.
and each of them can be labeled with a different color.
Okay.
Imagine you're watching a dance performance, but instead of just seeing the movements,
Right.
you can also see the emotions that each dancer is expressing.
Wow.
And their interactions with each other and their own individual stories.
So it's like going from a black and white film
Yeah,
to a vibrant, full-color masterpiece.
that's exactly right.
And by combining multicolor imaging with in vivo tracking,
Yes.
we could really start to unravel the complex choreography of life itself.
Exactly. We could see how different molecules are interacting with each other in real time inside a living organism.
This is incredible.
We could track how drugs are moving and targeting specific cells, or watch how immune cells are communicating with each other to fight off an infection.
Wow.
The possibilities are really endless.
This is all so incredibly exciting.
It is.
But as we wrap up our deep dive into this world of SMT, I want to take a moment to kind of reflect on what we've learned.
Yeah, I think that's a great idea.
We've seen how SMT is really revolutionizing the way that we understand this molecular world.
It is.
It's allowing us to see things that were once invisible
Right.
and to study these biological processes in a level of detail that we never thought possible.
Right.
We've explored all these incredible applications of SMT, from how proteins work to these virus-host interactions.
Yeah.
And we've even gotten a glimpse into this future
Yeah.
of in vivo tracking and multicolor imaging.
It's very exciting.
But maybe the most important takeaway from all of this is that SMT isn't just about the technology.
No.
It's about the people who are actually pushing these boundaries, the scientists who are asking these big questions and developing the tools to actually answer them.
That's what makes it so exciting.
It's a reminder that science is this constantly evolving journey, and it's driven by curiosity and creativity and this deep desire to understand the world around us.
Couldn't have said it better myself.
And who knows, maybe one of our listeners will be inspired to join this journey
I hope so.
and make some groundbreaking discoveries of their own.
That would be amazing.
So to all of our listeners out there, keep exploring, keep asking questions,
Yeah.
and keep diving deep into the mysteries of the universe.
I'll see you next time.